whatever. All right, we're about to get started, guys. If you haven't signed in, please get signed in. We're going to start here in just a minute. Once we do, this is our regular Kahoot that we're going to do to review chapter 7. So we'll be all caught up on all of our chapter Kahoots after this one, and then I'm going to jump into doing the projects. I'm going to run through them. I got to do 8 projects. You guys get to do 1 for the whole semester. So my projects, I want you guys to take them at face value as being the minimum, the bare minimum. None of mine are stellar, okay? So keep that in mind when you look at my project. They're what I consider base examples of what I would expect if you guys were using your own, because you guys just have to do 1 project for the whole semester. So look at it that way. My Kahoot quizzes that I made for this stuff are a little bit more, I don't know, you'll see what I mean when we get into it. I'm not going to go into too many detail on it. But give me what we usually do. I have a few different setups for that. I hope that you guys got my email saying that if you guys do need to email me for anything, please put your section on it. At least say Monday, Wednesday, AM. I don't need the number for it, right? But it is really hard for me to keep up with how many people have had emailing me about being sick or having accidents or having something that they have to be at, that they have to miss class. I have no idea what section they're in. I have to go through all of my sections individually and figure out what section they're in and like what days they're going to miss. Because they'll be like, what do I have to do to make things up? Well, if they're a Tuesday, Thursday person, and they would have to go to the lab before theirs on a Monday if they wanted to make that up. If they're a Monday person, then they'd have to go to the Tuesday one to make that up. And me getting all those details right. It's really hard to do if I don't know what section you're in. So please, if you do have any questions about anything, just like be like, hey, I'm in your Monday, Wednesday. Because I don't want to have to email you guys back and like, what section are you in? You know, to keep track of that stuff. So really would help me out a lot if you guys have that need. If you're just emailing about, hey, you know, what's a virus or something? Hopefully you're not emailing me about that, but hey, you know, what's this or that question that doesn't have any user section, and that's fine. No, cool. All right. Yes. What?
You must have
What did I do with those? Yeah, I don't know what that is. I'm imagining it's some sort of setting on there, but I don't.
Oh, never mind. It's
Okay.
Yeah, I got it.
I wish I had a better answer for you.
I think it's the numbers under it then. Your platform?
Yeah, I don't know. Probably some setting whenever we get to the questions. Okay, we're gonna go and get started. Whew. All right, chapter 7, traditional Kahoot that we're doing. Just to review chapter 7. True or false, antibiotics fight viruses. Something we'll definitely spend more time on in chapter 12, but we have mentioned this previously. Antibiotics only treat bacteria, period. Well, biotic would imply like anything biological, but it's not true. Antibiotics are actually antibacterial, and that's it. So that's the terminology for that. What are the drugs that we use for viruses? Antivirals. Okay. Everything else is pretty straightforward like that. Antifungals, antivirals, antiprotozoals. We even have anti-amoeba, specifically amoeba, anti-helminthics. My man, antibiotic is bacteria. Yep. So true or false, viruses enter host cells and use them to replicate, creating more viruses. Yeah, that was pretty straightforward. I feel like that one's not too bad. Basic. Why it's doing this weird pause thing. I hate it. Stresses me out when it does that. Okay, so true. Good job, guys. That's pretty much what they are. And that's why we don't really consider them to be, you know, alive. All right, what is the capsid around the viral genome made of? Oh, what the heck? I'm so sorry. The heck? Where'd my Kahoot go? What the heck? It's made out of protein. The capsid around the viral genome is made out of protein. All right. So this, we had a lot of people pick lipids. Y'all are thinking of the envelope. The envelope is a different thing than the capsid. The envelope is made up of a lipid bilayer. Envelope is different. All right. All viruses have capsid. All viruses have genome, but not all have an envelope. The viral genome, pick all that apply. Okay, in case, right. Why is it not the green one? Anybody want to say why?
Because they use another.
Because the virus will not have something green.
Right. It's sort of a trick choice, right? It says encodes the virus's cellular structures. Viruses are not cells. They can't have cellular structures. And organelles. Only eukaryotic cells have organelles. Viruses never have organelles. So that is there just to make sure that you guys are aware of what a virus really is and all that, but everything else is all correct. Good job. A little bit of trick choice there. Come on, I know, go away. Viral envelope. We kind of just mentioned this one. Go away. All right, great job. So this is going to be that outer membrane structure of some viruses. So it'll be outside of the capsid and the genome. Remember that structure of the genome and the capsid together is called the nucleocapsid. And sometimes that's all some viruses will need. Other ones might need an outer membrane on them. They are called enveloped viruses. Right. All right, icosahedral capsids in a virus. This is talking about the whole capsid structure. Right, so this one is the green one. So helical capsids are called helical capsids. They're not icosahedral. There's two kinds of capsids a virus can have, helical or icosahedral. Icosahedral means you have, yeah, it's 20. I don't care that you know that it's 20, but it's a bunch of triangle faces making up the structure of the whole thing. So the capsids themselves are not a triangle shape, but they have triangle facets on them. Okay, so that's an icosahedron. I'm not going to test you that it's 20 specifically. Bacteriophages, or viruses that infect bacteria, are involved in which of these processes? We did not talk about this like directly in the lecture, so this part that is on here, but yes, it is the term for what happens in this. All right, so the answer here is transduction. Okay, so when you are a bacteriophage virus and you're transferring your genes into a bacteria target and you're carrying new genes with you potentially and incorporating those genes in there and leading to lysogenic conversion, that stuff, that process is called transduction. That's something we're going to talk about in a little bit further future chapters. So this is not something that's going to be on your Unit 1 test, but it does relate to virology. So that's why this question's in here.
Sounds stupid, but if you remember adsorption, you can remember adjunction.
Yeah. Different ways to help you guys remember that. So lysogeny, what is that? In the viral life cycle, all right, so this is going to be when the viral genome inserts itself into the bacterial host chromosome. Okay, that's lysogeny. And again, if you are a bacteriophage that can switch between making viruses and just cranking out viruses as a product, like what we typically think of with a virus infection, right? You're making copies of yourself. You're replicating, reproducing. That's the lytic phase in the bacteriophage life cycle. So these terms only apply to bacteriophages, by the way. These terms do not apply to like human viral infection, only bacteriophages. So when a bacteria is getting infected with a virus that leads to more viruses, that's the lytic cycle. If the bacteriophage infects the bacteria and that virus incorporates itself into the bacterial genome, that becomes a prophage, then that is lysogeny. And bacteriophages that can switch between the two are called temperate phages, okay? So they can switch between those two. There's different ways you can induce them to switch, and we don't even know the details of how exactly that works, but that process would be called induction. Anyways, so we have lysogeny is that phase referring to when we have incorporated that virus into the bacterial genome. I remember that because it has gene in it, right? All right, lysogenic conversion. So specifically something that can happen as a result of lysogeny. You don't always have this from lysogeny, but it can happen. So this is the blue one. Most of us chose the yellow one. The yellow one says bacteriophage convert the bacterial genome into viral DNA. Do we convert the whole bacterial genome into viral DNA? No. Read that sentences and be sure that you understand what they're saying. So here we just talked about transduction as that introduction of the virus DNA into there. That whole process of becoming lysogenic is called transduction, okay? So we've gone through transduction. Again, transduction is not a term that will be on your Unit 1 exam, okay? We absolutely will be talking about that in this course though. So don't throw it away completely because you do need to know the term eventually. And it does apply to everything we already learned about bacteriophages, but transduction is that process of lysogenic conversion. And that's where we're going to be introducing new genes by inserting them into the genome. And we have a change in virulence. Anybody know what virulence is? The ability of that organism to cause disease. Okay. Also known as pathogenicity. All those terms are important, and the virulence and pathogenicity are going to be terms I'm going to throw around just assuming you know what they mean, just because of English language. So get ready for those terms, because to me, pathogenicity, pathogen, things like that are going to be such basic terminology, especially when you guys get into the healthcare field. They're not going to give you that as a term on your vocab list. They're just going to expect you to know what it means. So brush up on those and be aware of what they are going to represent in terms of diseases. All right, oncoviruses can cause blank in mammals. Most of us know this one. Yes, it is exactly like oncology. Onco, what's it mean? Cancer, exactly. So tumors, cancer, anything where our cells are growing unchecked. I'm going to wave around a sharp stick. Apparently, I'm already poking myself with it. And then we're going to get into our projects. First things first, for our first project, I have a handout. Grab one and then flip the rest back. Grab one and pass the rest. All right, so we're going to do our reviews of, again, my projects, my version of these projects, some basic variety, hopefully, in how we're going to be presenting this stuff. So our first one is going to be my handout. I'll have my handout up here on the screen as well as in your hands there at your desks. But basically, the first topic that I'm covering for Unit 1 is going to be our basics in microbiology as well as germ theory. So microbiology, as it says on our handout, it's the study of organisms too small. I need to set my timer, so it's going to do about 5 to 8 minutes. It's basically the study of organisms they too small to be seen with the naked eye, and so we would need to be able to observe them with a microscope or something like that. The two microbes that we talked about, two big groups of microbes that we talked about that are cellular, are going to be our eukaryotes and our eukaryotes. Eukaryotes that we also call prokaryotes. Those terms are interchangeable, right? So those are cellular guys. But what does cellular mean? Cells are basically membrane-bound
units that are capable of making copies of themselves. They have everything that they need inside of them to make those copies as well. So that's a cell. So we kind of have two things that we can divide up as cell versus acellular. And that would actually be our first approach. Cellular organisms, which includes our prokaryotes and eukaryotes, and our acellular organisms, which are our viruses and our prions. Okay. And so those are the guys that we would be separating that into, and then our cellular guys, the eukaryotes and the eukaryotes. So remember that the eukaryotes is going to be including our bacteria and our archaea. In that eukaryote and prokaryote refers to the fact that they do not have a true membrane-bound nucleus, and that these structures, that everything that happens in the cell all happens just within the free space of the cytoplasm. There's no membrane-bound structures, but they can still do a lot of the same functions that our eukaryotic cells do. But then our eukaryotics, those are basically going to be our guys that have membrane-bound organelles, including a true membrane-bound nucleus. And that in chapter 1, we just need to know that. And when we went into the different structures of the organelles, that was a chapter 5 thing, right? So anyways, these guys, again, the membrane-bound nucleus for the eukaryotes is where our genetic information, our DNA, our genome is stored. But bacteria have a genome too. They also have a DNA that they have, but it's just not in a nucleus. It's just floating out in their cytoplasm. But their genome is usually a big old circle, and that the eukaryotes have linear chromosomes, and usually multiple of them. For our acellular guys, the viruses and the prions, remember our viruses, they might have a genome in a protein coating at the bare minimum. Some of them have an envelope surrounding them. It's the stolen lipid bilayer that they stole from the host cell that they had infected. And then our prions are just misfolded proteins. And those misfolded proteins get introduced into the body and cause the healthy proteins to misfold, then aggregate in tissues and cause damage as a result. So that's those guys. One of the other topics that we talked about basically was the introduction of what disease even is. Disease is any perceived deviation from what you think is your norm, right? So what you're used to every day. So just because you wake up with a kind of a prick in your neck or something, I mean, yeah, we could technically call that disease, but is it really a deviation? It's probably you know what's going on with that and all that stuff. So once we have that progressive sort of development of that deviation from health, we can start seeing signs and symptoms. That's going to be disease, and what disease would be defined as. Pathogens are any microbes that would lead to the development of disease. So if your disease is caused by a microbe or any other organism that's introduced in your body leading to that disease, that deviation from health, that's a pathogen. And so we talked about our different groups of pathogens as potentially being virus, prion, various bacteria. They could fall into gram-positive and gram-negative, or the ones that don't have a cell wall at all, are mycoplasma, or the ones that are modified with the wax or mycobacterium. That we could even have archaea that can cause disease, right? We've talked about already with tooth decay and things like that. Archaea can contribute to that. With our eukaryotes, we have our fungi, that includes our yeasts and our molds. We have our protozoa, and we have our helminths, that those guys can all be contributors to disease as well. Back in the day, whenever they didn't really understand what caused disease, they just believed that it came up out of nowhere. And so the whole idea was that disease, as well as when food spoiled or anything like that, that it just, life could just come out of nothing. And that was called spontaneous generation, that life could just literally just spontaneously exist. As God willed it, if you can imagine. That's how they imagined it to be. But as we have learned, life begets life. And so we just couldn't see those life forms that were already there being introduced through the air or from the environment, somehow contaminating the things and then overgrowing as a response, being on their nutritious media, basically. Even old meat and things like that serve as media for them. Louis Pasteur came up with his basically his design of this experiment that would lead to the development of germ theory, which is the theory that germs are causing these things, that we have existing life in these environments, and that germs are the things that are contributing to that. In our case, we're talking about the microbes, right? Anything that would be too small to see with the naked eye. But he took, basically a sterile media flask that was open and a sterile media flask that had impossible for bacteria just to get into it, but we can still get air in there. So these guys still need air, just like we do. And so he did that, and he showed that if he had that contained thing that was hard for microbes to get into, that it didn't grow anything. But if spontaneous generation was the explanation for everything, it should have grown something, right? Certainly God can will for something to just grow in that. If, how's he going to be impeded by the fact that there's a wiry little neck on it. And so that was his experiment that he did to try to introduce that theory of germ theory. And that was Louis Pasteur. All right, so that is our review of chapter 1. Now, something that I want to demonstrate for you guys. Anybody that might have been watching, I know most of you guys are sitting there wishing you're dead listening to me talk, but anybody that was watching me give my review of this handout, how many times did I look at the handout? Was I staring at it, reading off of it the whole time?
No.
Right, did I stand up here and talk about the information? You only use this as a reference point to make sure I was hitting my points. That's how you guys have to give your presentation. You're reading off of your pamphlet. I reserve the right to ask you questions. If you're standing up here talking and you're showing me that you learned the info on your pamphlet or your slide or whatever it is, then I probably won't ask you questions. I'm going to assume you know your stuff. But if you're like microbiology, the study of organisms too small to be seen, that sort of a thing, I'm going to reserve the right after you're done to ask you questions. So I'm going to say, put your handout down, close your screen. I've got some questions for you, and see if you can actually answer those questions for me from your brain and not just reading off of here. Because believe me when I tell you that became a problem last semester. That we had groups doing these things. And there were people, somebody might have made the whole thing for the whole group, and we would just have people come up and read the question and say, yep, this is the right answer off of the Kahoots that they would give. And that pissed me off. And anybody that was in that section would tell you I was pissed off about that. I made it pretty clear I was pissed off. So if you're gonna come up here and expect me to give you credit for having done your work, I expect you to show me that you did the work and you didn't just like copy and paste some stuff or let, like you guys don't have groups. Part of the reason why I got rid of groups, because one person can carry the whole group that way. And if I were to be like, hey, you know, for the whole group, somebody answer me this question. Obviously, the person that put that quiz together probably would have been able to answer the question. But the person that was reading the question and gave me the answers, how many times do you think that they were able to answer the questions that I asked them about that? Only like one person was able to. Of all their Kahoot questions, they had like 10 or something questions on there. And they couldn't even tell me what the heck they were asking in their questions once I turned it off. That's disgraceful. And you guys know the projects aren't about you putting up a thing and reading it directly off of it. It's about showing me that you understood the topic that you were assigned. And me coming up here, you could have made it the night before and come up here and told me all about the thing you made the night before. I'm not asking you to put in like, you know, that kind of effort, but, and that even that's sad, I feel like, to put it that way, but just so that we're clear, I expect you to know your topic. And then I reserve the right to ask you questions about it to prove that you know your topic, okay? All right, moving on. Obviously, I know the topic, but you guys will have to know it too, all right? Okay. We're not ready for that yet. We gotta,
Yeah.
it's coming, believe me. Oh, it's coming. So the next thing we're gonna do is our 5 eyes. How do I put this on full screen? Here we go. All right. So for our 5 eyes, this was a chapter 3 concept. So for chapter 3, these are our 5 eyes, and I put the little thing up here for our reference so we can start talking about those 5 eyes. And if you didn't notice, also I was timing myself. Before I started getting into the whole thing of like, make sure you know your stuff. Did you see how many times? That was around 6 minutes, okay? So that's not very long. That's all you have to do it for, okay? So starting my timer for this one, the 5 eyes. The 5 eyes are the major steps involved in identifying microbes, basically based on a sample that you may have collected that could have a variety of microbes in it. But how do you get to the microbe that you're interested in? How do you figure out what that microbe is? Well, this image here is a great way to refer back to refresh you guys' memories on the 5 eyes. So if you guys are looking to study up about the 5 eyes and what each of those eyes really are for each step, this is a good one to come back to for your review. But we're going to go on. Our first step in the 5 eyes is inoculation. This is introducing your sample, basically the swab of the throat or the wound, or whatever it could be, onto sterile media. And that's all inoculation is. The actual getting it to grow part, that's incubation. Just putting it on the media, that's going to be our inoculation step, okay? So our media needs to be sterile. It needs to be completely free of any living organisms whatsoever. That's what sterile will be. For inoculation, again, that's just literally putting your sample onto the sterile media. That's the practice of doing that specifically. Our medium is singular, media is plural. Our medium is whatever that material is that you'll be growing your microbes on or in. It could be liquid, it could be solid, and it could be semi-solid, so stuff in between and what have you. But no matter what, it's providing the nutrients in order for your microbe to grow. And then the culture is basically what results as after you have grown your stuff on or in the media. And so that's the idea of inoculation. For incubation, this could go away. Let me see, come on. All right. For incubation, this is essentially putting your inoculated media at appropriate temperatures, amount of oxygen that they need, or lack of oxygen if they don't grow well in oxygen, if you suspect that they can't. Putting them at the right condition to encourage them to grow up in your sample. So here we have a table at the top left that's showing us some common microbes and their optimum growth and culturing time. So with listeria, which is a common contaminant of food that can lead to foodborne illness, its optimum growth temperature is 25 to 30 degrees Celsius. So just a little bit warmer than room temperature. So here we're talking about like 80 degrees Fahrenheit, culturing time about 1 to 2 days. Or if you scroll all the way down to Mycobacterium tuberculosis, it prefers to grow at 37 degrees Celsius, that's body temperature, so 98.6 degrees Fahrenheit. And its culturing time is 28 days. So it takes 28 days in order to have like an actual usable culture for that guy. So an entire month just to culture it, which is why we have so many tests available for tuberculosis that don't rely on culturing, because you're just not going to get it if we have to culture it. So when you're actually incubating these guys and inoculating these guys, this is what will get us to our ideas of the different types of media we can have. We can have general purpose media, just general use. Most things should grow on it. So you can, if you don't have any idea what your microbe could be, you would want to start out with a general purpose media, and then maybe move from there based on what you might find from your gram stains or whatever you might have. Enriched media has a growth factor added to it so that fastidious bacteria can grow on it. Remember, those three terms always are going to go together. So a growth factor is something that that organism can't make for itself. If an organism can't make something for itself and it needs it in its diet or its nutrition, then we call it fastidious. And so that media that has the growth factor, we call enriched. Those three things go together. We also introduced selective and differential media. Selective literally has something in it to prohibit growth from some organisms, therefore favoring growth of others. So selecting for things. And then we have differential media, where we can basically tell the difference between microbes, usually based on a color change in the media or in the growth itself, from chemicals reacting with like metabolic byproducts. So wastes that these guys make cause the media to change colors, and then we can kind of tell differences based on that, if you can ferment or if you can do other things. My image apparently disappeared from this box on here, but that's okay. It was basically talking about what a colony was. So isolation is our next step, our third I in the 5 I's. Isolation is separating out that mixture of bacteria and then targeting your microbe of interest. And so the way that we can spread those out, the way that we learned in our lab, right, and the way that I'm going to test you over on the test is going to be the quad straight plate. So here we have an image that basically walks us through those steps. Area 1, you place your stuff down, flame your loop. Area 2, we're spreading out what we picked up from area 1. Flame your loop. Area 3, we're spreading out what we pick up from area 2, and so on and so forth. So it's really important that we remember that we have that sterilization in between each of those streaks on the quad plate, so that you understand the purpose of the quad plate, which is just to get those individual cells spread out. And an individual cell of bacteria should grow up on itself and lead to a colony. So a colony represents one cell of bacteria that has grown up into a population of bacteria from that cell. They're all identical to one another as a result. And so you could literally take a sterile instrument, pick one of those colonies, and create a new culture in sterile media. And that would be a pure culture. And that's the whole point of the isolation step and what this image here kind of depicts a little bit. All right, our fourth step in the 5 I's is inspection. We're already at 5.5 minutes to show you guys how quick I expect your presentations to be. Using biochemical or metabolic phenotypic tests, immunologic antibody antigen tests, and genotypic tests. So basically using test results to get data. That's what the inspection step is. This is the collection of the data. And then I have a little bit of breakdown of each of these here on the slide, as well as our definition of antibodies and antigens for those of us who don't know what those are. And so it's basically inspection step, collecting the data, and then identification, using all the data that we collected to determine the identity of your microbe. This could be using dichotomous keys, which would just basically have a choice between two things. Did it do this thing or this thing? Yes or no? And depending on if it did or didn't, go down that path or the other path. Okay, so that's the idea. And then just break it down into two choices after that and so on and so forth from there. Then we have Bergey's Manual of Bacteriology, which is just a literal book or an online site. You can get it either way. It's a compendium of all that data that you can look through to determine the identity of your organisms. Anybody have any questions about any of that? All right, so we had a lot of, yes.
How does the compendium decide, like, ID those?
So you would literally like, if you like, okay, well, I know I start off with, I have all this data, right? And I know it's gram positive. Then you go into the book and like look up gram positive and then find like another test that you did. Okay, it was able to ferment glucose, and then it would tell you to go to this page, and then it like puts you like physically, it's like different books. It's a pain in the butt to get to the physical books, but the online site kind of does that work for you. So yeah, we won't use Bergey's manual, but we will use dichotomous keys in our lab, and I'll make a dichotomous key for you guys to follow through what our results were. Any other questions? All right. So if you guys decide to do PowerPoint slides, which most people do, you are welcome to put a lot of information. So you guys saw me on this slide here, where I said, here's some slide with some information. Basically, I told you what inspection was, and I had a little bit more in-depth information on my slide. You guys have 5 to 8 minutes to cover it, but this counts within that time and that information. So if you have it on your slide and you just summarize it, but it's there on the slide, that counts as covering the information. But I want you to keep the summary of your slides within that 5 to 8 minutes, okay? All right, that's 8 minutes. Next topic is the gram. I think I had the gram thing. I know it's supposed to be the song. Let's do the song. Fine, we'll do the song. So we'll do the song. Guys, this is not me showcasing my singing ability. I want to be clear. This is not me trying to show off. No, I don't sing. Like, that's not a thing I ever did. I did a song just to help you encourage you guys to be more creative with your stuff, okay? So feel free to laugh at it. It's meant to be cheesy. It's meant to be a little cringe. It's okay, all right? Just don't take it too seriously. This is not me trying to win some kind of singing award. I can't even watch it, like, or I'm gonna turn beet red if I watch it. This is the most embarrassing thing I've done in a long time.
When did you make this?
On Saturday. I think Saturday, it might have been Friday, I don't really remember. Oh no. I'm gonna sing.
English is the thing that dictates all living things in this world. It has a lot to do with how proteins fold and how molecules and cells work. Whoa, whoa, here's just high, or here's just low, and the things in your cell just won't work anymore. Yeah, it'll take a huge toll on you, and soon you won't be you if you can't regain that control. Don't you know? We ain't home. That's all I want to do.
I know, this is so good, isn't it?
Yeah,
Like I said,
I'm
I'm not trying to win awards.
That's all I want to do. About 7 hours all night, that means to go through mathematics, the inverse of the proton concentration. Big twist of pH, goes 10 to -2, we pH 2, and that means more acidic than what pH 5 says. Mathematics, water's made of H2O, but it's not static. H2O falls apart.
Right, not static.
And
Dictates that.
in equal amounts, and that pH 7 is true.
Yeah, it's okay. Laugh. So, I didn't realize that the music wasn't recording and it didn't play in the background. So when I watched this video and it got to the guitar breakdown, I was like rocking out like I was playing to the guitar. And then there was like no sound. So I did something else instead. So hopefully you guys enjoy what I did instead.
Good, like her stuff.
If you guys decide to do a song or something like this, please feel free to be silly, be stupid with it. Get that information in there, and that's what's important, okay?
I love it.
Yeah.
Those kind of
Yeah.
video background music.
Yeah, so I could have like added in, I guess, the track back over this and
No,
like
I
had the
wouldn't.
instrumental in there, but I was just like,
Okay.
yeah. Wanted the info in there. The info obviously is more important. And you guys are welcome to use whatever songs you like, even if the song that you guys are doing your lyrics over has profanity in it. And you know, obviously I'm not gonna be too strict on the profanity thing, and we're all adults in here, but you're welcome to change the lyrics to suit whatever your needs will. So if otherwise it might not be an appropriate song, to change the lyrics, make it appropriate. So
Good luck on that. It's gonna be stuck in my head all day.
yeah, so there you go. You learned about pH to Teddy swims.
You want which house?
So yeah, so this is something like make it a
Have to go
skit,
there.
make a music video out of it and whatever. Like I literally just sat down in front of my computer and turned it into a 3.5 minute song and wrote some lyrics. It's super cheesy, but it's a lot more fun than the slides, right? So that's the idea. So the next thing I have is my gram positive and gram negative model. Again, this is not exactly like, I meant to make a better model, but I couldn't get the glue to stick to I had like glass and I had balloon in the glass and then on the outside, but it wouldn't stick to the glass. And then I tried it with the styrofoam and the glue wouldn't stick to that. So again, this is the whole point is that we understand the difference between gram positive and gram negative. So we're gonna focus on that. That's what's important, right? All right, so gram positive and gram negative cells. So we learned that bacteria have like the whole outside of them. They can have membrane, walls, and outer membrane. All of that together, that outer structure is called the cell envelope. And that all bacteria cells have a cytoplasmic membrane. That's the membrane that contains the goo on the inside. So it's the inner membrane, the cytoplasmic membrane. Almost all bacteria have a cell wall of some kind. We learned 2 exceptions that kind of break the rule of the peptidoglycan walls, right? The ones that don't have a cell wall, which are mycoplasma, and the ones that have a cell wall, but it's modified with the wax mycolic acid, and that's the mycobacterium, right? And everybody else is either true gram positive or true gram negative. And then our gram positives have that thick peptidoglycan layer that gives them a lot of structure and support that inner membrane kind of attaches to. That thick peptidoglycan layer is supported by lipoteichoic and teichoic acids. Helps with the maintenance whenever we're making copies of the cells and things like that, as well as with the overall structure of those cells. Whereas the gram negatives have an inner membrane, and then they have a thin cell wall made of peptidoglycan, and then an outer membrane. And that outer membrane on the gram negatives is studied with lipopolysaccharide, which is AKA LPS, AKA endotoxin. It's all the same thing, okay? And that's the general makeup of those guys. And the actual makeup and structure of those cells will kind of dictate how they behave with the gram stain as well. So I have little models here, and they did not have purple at the dollar store. And the red was like super, super dark red, and I didn't really like that. So we get blue and pink. So it's a boy and girl, I guess. I don't know. There's some traditions of, yeah, traditional societal rules, I guess. So, right. So our gram positive and our gram negatives. So our gram positive has this thick, the white is our cell walls for both of them. Okay. So the white is the cell walls for both of these guys. Here, our gram positive has our thick wall here. It doesn't have an outer membrane, right? So it just has the wall, but it's a thick wall surrounding the cytoplasmic membrane, which is our balloons. So our balloons are our cytoplasmic membrane. Imagine that inside of the bacteria, we have everything, all that goo, our genome, and everything that happens in the cell contained within the cytoplasmic membrane and in the wall on the outside. So it's obvious to see this set up this way, that this one's going to be sturdier than this one. Here, our cytoplasmic membrane with our thin wall, which is our paper towel here. And I have crepe paper or creepy paper, if you know, you know. So the crepe paper here surrounding these, that is our outer membrane here. Okay? And this would be studied with lipopolysaccharide and whatever. So obviously, if we put these cells into salt, that's going to cause all of the water to leach out of these guys. And what's inside of these guys is that cytoplasm, which is primarily water. And so if we start, you know, packaging food in cells, or if we're growing on our skin, which is salty, it's even slightly salty environment, we'll start to lose that water out of our actual cell membrane, and that'll cause that everything that's in there to shrink down. And basically, whenever we go through that shrinking down, The gram positives, which attach itself membrane to the actual inside of that wall, are going to be able to maintain their structure better because the cell wall is sturdier than with the gram negatives. And of course, what am I going to do? I'm going to pop some balloons. Don't everybody have a heart attack at once. Okay, so gram positives, we put it into salt, the inside shrinks, and we'll still maintain the shape of that cell, still nice and sturdy. Whereas with our gram negatives, it's not going to maintain that structure as well. And so that can be deadly to the cell. And this process is called plasmolysis, when the cell shrinks down like that as a result of their environment. So that demonstrates the structure that that cell wall would give. Does that make sense? Okay. So that was 4 minutes. If you guys, I will say this too. So on this, I gave a little explanation because me just like being like, oh, I'm going to do a model and just hoping that's going to be good enough. I gave my explanations, what I submitted on here, so that if an instructor was grading this, they would know what I was going to be talking about and know that I have like my stuff together beforehand. And I explained how I was going to use the model, and I even put pictures of the model and how it was going to work on there. You could attach a video if you wanted to, so that I could grade it and like give you guys critique. So if you guys submit your stuff early, like even if it's just a few days early, it gives me time to look at it and feel like you might want to include this, that, and the other, or you might want to add this term too, or something like that, right? So you guys can submit that stuff early enough, and I can give you feedback if you give me time, okay? So I have that stuff on there so that people could see this online and still understand about what it was about. But obviously doing it in person makes more sense. But if you guys are able to get the information out, like I reviewed gram positive and gram negative cells in under 5 minutes, you still are going to get full credit as long as you cover all of your information, okay? You don't have to keep it at 5, it's just a recommendation. Please don't go over 8, because I just can't accommodate everybody if we go over 8. All right, cool. So we did this, get out of the way. We did this. So now we're on to the Kahoot quizzes. Oh my God. Please go away. I need to sneak up to it, I guess. Here we go. All right. Let's do this. Your folders, this, projects. Yes. Okay. So we'll do our bacterial structures and functions. I wanted to do all of the Kahoots all at once. So we're going to do Kahoots. The next 3 are all Kahoots. And they're different formats of Kahoots, okay? So the first ones are traditional Kahoot. You guys know what to expect from this. So we're just going to go through this just like we traditionally would. Everybody join in.
Loading thing.
I'll turn the sound up so it's fancier. And you guys are welcome to do Kahoots as well. What you're able to do with Kahoots on your own for free is limited. So if you want more features, if you want to do Kahoot for your project, if you want more features, let me know and I'll give you a pass to use my account. Okay, and you'll be able to do all my features. If you want to do Kahoots, there's a caveat with Kahoots that people learned last semester, which is part of the reason why I got pissy. If you're going to put up 10 questions in your 5 minutes about your info, your questions still have to cover everything that is on that study sheet, right? So sometimes that would mean not just explaining the right answer, but being able to explain the right and the wrong answers, right? You still have to cover all of your information. All right, we're going to go ahead and get started. Again, this is not for your credit or anything, it's just for you guys for practicing it. So if this was my presentation, I would still go through my questions as well as my answers. Oh, right. Okay, I forgot there's a poll on here. How often do you think about bacteria? I forgot about this. If you're me, then you probably think about it all the time. I think about it a lot, man. That doesn't mean I'm as good about washing my hands as I should be. All right, which structure aids bacterial motility? So here, you can see how this question, how does it aid motility? And I have these choices. It gives me the opportunity to talk about the other structures as well. Okay. All right, so flagella aid in bacterial motility. The capsule is just a sticky coating that we can use for adherence and evading phagocytosis, creating biofilms. The pili, that creates a channel between the cells that allows us to move DNA between 2 cells. It can be used for attachment as well. And the fimbriate is really mostly only used for attachment. So flagella, these are our modal structures. All right, true or false? Bacteria and archaea lack internal organelles, including the nucleus. Is true or false? All right, so this one is true. They do not have internal organelles, or membrane-bound organelles. They lack those, and they do not have a nucleus. They still have genetic information. That's not the same, right? The nucleus is the structure that contains that. All right, next question. Complete the sentence. The glycocalyx can form as. All right, so the glycocalyx is the capsules and the slime layers, those sugary coatings. Glyco referring to sugars, calyx coating. If we were going to go back to the question again, the cell walls and membranes, again, those are going to be part of the cellular envelope, not the glycocalyx. That's a separate structure. Flagella, motility pili, connecting and transferring things between cells, whereas the fimbriae pili for attachment, and again, pili transferring between cells. All right, which shape is rod-shaped? Which cell shape is the rod-shaped one?
Sounds terrible, but I do this every other time.
Yeah.
My high school teacher taught me that, and she's like, "It looks terrible, but I worked at NASA, so it works."
It does work. All right, so the bacilli, great job. So spirilla, spiral shaped, cocci, spherical, pleomorphic, doesn't have a set defined shape, right? All right, which bacteria form endospores? All right, so our endospore forming bacterium are always going to be Bacillus and Clostridium. And that'll be their names. It'll be Bacillus something or Clostridium something. So over here we have something called Lactobacillus. That could potentially, has bacillus in its name, it could potentially be endospore forming, but Bifidobacterium, that doesn't equate, does it? So that one is not our answer. Nobody picked that one, but yeah. So endospore forming bacteria is always going to be called Bacillus or Clostridium. All right, what is a tuft of flagella called?
With the study, because are we allowed to do it multiple times? Okay.
You just need to change your
I've
username.
been having it, I have done it like 2 times and just think. So I'm just
Yeah,
gonna
that's
start
fine.
doing stupid ones.
Yeah, that's fine. I don't track that stuff, so you're good.
I was about to say, I just wanted to make sure, because I'm about to do stupid names.
You're good. All right, so tuft of flagella, again, this is a flashcard term type of thing, right? Lophotrichus, amphitrichus at each end, peritrichus all around the whole cell, monotrichus, just one flagellum. Those are our 4 terms for our flagella. And if this was your Kahoot quiz that you were presenting, I would expect you to explain the other ones. Which is the process of forming an endospore? What's the term for forming the endospore?
Oh shit.
Oh, I don't have any. Not here anyways, in my office.
My tattoos finally like completely healing.
It gets so itchy, yeah. All right, I always use Aquaphor. All right, process of forming an endospore, sporulation. Germination is going from the spore to the vegetative. Transcription is going from DNA to RNA. Replication is just making copies of things in a reproductive kind of way. All righty, what are glycocalyx functions? All right, so our glycocalyx functions. Remember, glycocalyx, sugar coating. This includes our capsules and our slime layers. This is for adhering to surfaces, evading phagocytosis, and forming biofilms. When we look at our other choices, photosynthesis, respiration, fermentation, this is mostly going to be occurring in our mitochondria and our chloroplasts. Our motility, replication, transcription, those are completely different things. So that's not going to occur with just one particular thing. Protein synthesis, DNA repair, and cell division, kind of relating to making copies of the cell, but that's not what the glycocalyx does for us. That's our sugary coating. All right, question 10. Which of the arrangements describes irregular cluster? All right, so sorry, click past it, but that's okay. We'll go back to it. Staphylo. Staphylo represents the regular clusters. Strepto, strip, chains. Okay. Palisades, that's our fence posts next to each other, and our tetrads is groups of 4. So those are not irregular clusters, right? Those would be very regular. True or false? Endospores are metabolically active. True or false? All right, so this one is false. Endospores are not metabolically active. They are dormant. They're completely inert. If they want to become back to metabolically active, dividing, to doing things, they have to be vegetative to do that. Endospores are not vegetative. They cycle between those two. All right, question 12, complete the sentence. Bacterial cell envelopes. What's the best option here? Be sure that we're actually understanding the choices. All right, great job. So we got this one. A little bit tricky here. Again, the bacterial cell envelope will include the outer membrane if you have one, right? But it's not only the cell wall. It's not only the cytoplasmic membrane. It's the cytoplasmic membrane, the cell wall, and the outer membrane, if you have all of those things, right? So it's all of those things. The cell envelope does not include the glycocalyx. Most things will only make those under certain conditions. All right, true or false, archaea have peptidoglycan in their cell walls. Looks like little like beanie weenies. All right, yeah, like exactly like Vienna sausages is what it looks like. All right, true or false, archaea have peptidoglycan in their cell walls. This is false. They have a weird molecule, man, and I don't even know what, we don't need to worry about what it is, but it's not peptidoglycan. They also don't have phospholipids in their membranes. They have weird molecules. So they're just weird.
Aren't they naked?
They do have membranes. They do have that, but it's just not made of phospholipids. All right, what is the function of pili? We only got 2 more questions left. Function of pili. For time a little bit. Pili for attachment and conjugation. All right, conjugation is transferring DNA between the 2 cells. All righty, last question, double points. Which bacteria have variable shapes? All right, so this is our pleomorphic. This is what this means. Pleo means many, morph means shape. So good job, guys. Here's our podium for this one. Then we have another Kahoot after this one. We're going to do it a little differently. Good job, good job. There it is. Couldn't see my cursor. Alrighty, our next one. I should have done this one first. Dang it. Okay, so we're gonna do this one the other way. That's all right. I meant to do this one as our group competition, but that's okay. All right, so let's do two. It says 1 to 3, but it won't let me. It won't let me take away groups. All right, there we go. So we'll have 2 groups. So you guys have to join Team Horse or Team Brain. Horse or brain, because these things are very related to one another.
I was with Ramsey originally, so I agree with you.
Oh, yeah,
You don't need our actual names, so I was like, I'll just do Ramsey.
that's fine. Yeah, I don't need your actual names for this, guys. All right, so we're gonna go ahead and get started because I don't want to lag too much with the timing on these. All right, so this is your teams. So keep up with your teams. If you need to join, there will be a, if you didn't get to join in the beginning, there's a code for this still. All right, which of the 4 types of macromolecules would antibodies or enzymes fall under? Here's our pin down here at the bottom if you needed to join later.
Would it fall under either proteins or nucleic acids?
Yeah, if you guys want to work together as teams, you know who's on your teams. You could shout out. You might be helping out the other team. So one thing you guys are welcome to do, if you guys offer a team thing, you are welcome to ask the class to like sort by teams if you want to. I just didn't want, I could have said like this is team whatever and that team whatever, but like I didn't want to deal with it right now. But when if you guys decide to do team competitions, you can. All right, so here's our results. All right, so our answer for that one, just to show it, that's proteins. So enzymes and antibodies fall under proteins. All right, next question. What is the monomer subunit that makes up carbohydrate? Oh, why it takes so long, I guess, to let you guys read the question, I guess. You guys got this?
I got you, bro. You can have them all. I have plenty of them. Thank you. I need salt.
All righty, so that's saccharides. We're doing pretty good. We're doing pretty good. Next question. You can select more than one. What are nucleotides? Be sure you read your choices here. Yeah, it allows for team talk, basically. So next time I do this team thing, I'll be sure that I let you guys be in your specific areas.
It's the makeup of DNA.
Right, so that's fair enough.
That helps you out.
Right, so we have 2 of these are correct. It is a 5-carbon sugar, phosphate, and nitrogenous base. Then monomers for nucleic acids. Why is red not correct? Because ribose is found in RNA. It is not found in DNA. Why is the blue not correct? Nitrogenous sugar. The sugar is not nitrogenous. The base is. The sugar is 5-carbon. The green is not correct because it's not a 6-carbon. It's a 5-carbon or a pentose. And then the purple is not correct because these are not monomers for proteins. They are monomers for nucleic acids. Next question. True or false? Phospholipids have hydrophobic qualities.
I mean, they're emphatic. So, yeah.
Yeah.
Their tails. I think that is no. Their tails are non-polar, correct?
Correct.
So they are. water's non-polar, right? No, water's polar,
Mhm.
meaning the non-polar tails are hydrophobic. While the heads are.
All right, so you guys did great. So this one's true. It does have hydrophobic qualities. It has both hydrophobic and hydrophilic because they're amphipathic. I heard you guys talking about that. Great job. You guys are getting that. Next question. It's a puzzle. Using descriptions of protein structure levels, place them in order from primary at the top to quaternary at the bottom. So it is kind of in the picture, okay? Not all of it is, but some of it is. But you need to be able to read your choices and put them in order from top to bottom. Primary is at the top, quaternary at the bottom.
High school, my sophomore semester year, I had biology, and we had to do this puzzle daily until
That sounds like fun.
it was.
It would be very discouraging. I was in jazz band. I was in one jazz band, and I had to like play. We had to play all like the scales, major and minor, for every like letter in the music scale. And so that, of course, took most people days. But then our guitarist just sits down and does all of them all at once. And yeah, it made me feel stupid because I played saxophone, and it took a lot of work to get all of them. Anyways, so top, primary, amino acid sequence itself. Second, the R groups next to each other. So those things that make the amino acids, the different amino acids, those R groups next to each other make the alpha helices and beta sheets. Second, the tertiary shape, that's going to be due to R groups far apart, but we're still in the same ribbon, right? And then bring multiple polypeptides or multiple ribbons together to make functional units. That is our quaternary. All right, so that's where we got. 8 of you got it right and 13 of you got it wrong. All right, if most sugars are based around the formula C6H12O6 or something similar, do you think sugars are polar or non-polar? We've talked about this previously, but see if you guys can remember.
I am your skeletons are all non-polar, except for those little opposites. Yeah, those are the polar. That was my favorite thing in biochem.
Wrong. It might have a lot of carbons and hydrogens in it, but we have OHs scattered all throughout those suckers. So let me ask you guys, when you guys put sugar in water, does it dissolve? Easy, easy. It's polar, easy.
Well,
Y'all thinking way too hard about this. It's got all these C's and H's, yeah, but look how many oxygens we have. Polar, okay? And those polars are scattered all throughout those oxygens, okay? Sugar dissolves really well in water because it's polar. It's okay, that's all right. Just be sure you get all right on the test, right? What is the defining chemical characteristic of lipid? Lipids, which one applies?
Does a piece of wax dissolve in water? Everything I say is satire.
Yeah, you're fine. You're right. I have to check that because I'm a professor, but when in my real life, I'm very
Oh,
sarcastic.
you
Just not allowed to
you
do it
can
as much.
to me, I will.
So we have the characteristics of lipids. Lipids are always have a hydrophobic characteristic. Now, they can be amphipathic, but they're not always amphipathic. So that's why the yellow is not correct for this one. They're not always amphipathic. They can be. All right, question 8. Pick all that apply. All of your genes code for proteins, or they'll control when proteins are made. What functions do proteins have? We actually talked about some of this a little bit, but it's a lot. So pick all that apply. Lucky the test doesn't have select all that apply, because this would screw a lot of people over. So it's all of them, okay? It's all of them. It's all of them. So y'all that are saying you've never heard of main catalysts, you apparently have not been paying attention to what an enzyme is. An enzyme is an organic catalyst, okay? So it helps overcome activation energy in our cellular processes. We're going to talk about that in chapter 10 in a lot of detail, okay? But eventually we'll get to all of these functions of the proteins, but this is just reminding you guys, proteins have a lot more function than probably what you realize. All righty. Question 9, true or false? The most important energy source in all of our cells falls within nucleic acid macromolecule grouping. Energy source. So our energy source for cells, what grouping would it fall under? Is it nucleic acid? Say we have a suggestion from one group. What group were you in? Horse or brain? Sorry, I'm not looking at you at all, Jordan.
Oh, I'm in Hans.
So if you guys were in horse, we have a suggestion that's ATP. Is that nucleic acid? So it is a nucleic acid, adenosine triphosphate. Okay, so that adenosine with the phosphate, it's also got a ribose with it. It's actually a nucleic acid. It's just got 2 more extra phosphate tacked onto it. Okay. All righty. This is it. Are we close? 10. Only 2 more questions left. This is number 10. Multi-select again. Which of the biological macromolecule groupings are considered organic molecules? Which of these are the organic molecules?
You just want to say carbon. That would be very wrong.
So yeah, think about what an organic molecule is. What do you need in order to be organic? Carbons, and they usually are bound to hydrogen somewhere. So it's all of them. So I think most of you guys got that. Yeah, it's all of them. They all have carbons and hydrogens. All right, true or false? The way biological macromolecules interact with water majorly impacts structure and function of our cells. True or false? Those macromolecules, carbohydrates, proteins, lipids, and nucleic acids, how they interact with water impacts our cell structure. Yeah, so true. Obviously, that's true because we talk about the polarity and the non-polar and all that. So I'm not going to have time to do all of them. We're going to do one more Kahoot. Okay, it's going to be a different format than these. And then we're not going to do the virus whiteboard thing. We did just do chapter 7 though. So if you guys want to review the virus stuff, I have the handout that have been used for the whiteboard things. But what I intended to do was literally just review it actively live. But the whiteboard content is on the handout posted in that discussion group if you want to see it. But we're going to do one last Kahoot game because it's more fun than watching me talk. I mean, can we agree on that at least? And then, all right. So our last Kahoot is going to be our eukaryotes. And this one is a discussion-based one. It's all open-ended questions. I know, suck and also good at the same time. I don't really know. All right. So we'll do this classic. All right, everybody get logged in real quick so we can crank through. You can use whatever name you want to use. It doesn't have to be your actual name if you want to use. You want to be Gordon Ramsay there. You want to be Beyonce. You be whoever you want to be right now. Like I said, if you guys want to review viruses, I do have that posted in the discussion post. I'm going to show you really quick. If you go to discussions and click on unit 1 project examples, scroll down, and in the replies, I have everything. You would just click over here to see all that content that I would have covered for viruses. We mostly covered it in the Kahoot that we had, so we should be good. All right, we ready? Question 1. These are different formats than what we have been doing. How do flagella, cilia, and pseudopodia differ in structure and function among various eukaryotic organisms? Again, basic, I don't need like full sentences or something. You're not getting actually graded on this. I don't need a novel. We might skip a question or two on this specifically because I don't know if we have the time for all the questions, but we'll try to do the best we can. Yeah, just click a random one. It's not a big deal.
Oh, I had to click over.
It's just gonna help us sort of organize the choices. Okay. Mobility, not wrong. Okay. So not wrong. So, right. So we've got here flagella extensions that help with movement, cilia, protection of cell things and moving things across the surface. Right. So cilia kind of a little bit protect, not their main function. They can move the things around the cell. They can technically also move the cells too, but they're tiny little hair-like guys. The flagella are like long projections. And then the pseudopods are false feet. So this is literally pushing out the cell and moving around the whole cell that way. So we have some selections based on that we could go through here and see these things. Right, so here we have mention of the flagella can run and tumble. That is the case only for bacteria. So eukaryotic cells don't run and tumble. They can actually direct their movement with their flagella because their flagella can move like whips. They don't rotate and switch. So this is talking about eukaryotic organisms. So we get an idea of the differences there. Another question, what evidence, actually skip this one. We're gonna skip it, just don't worry about it, because guys, be aware that there's evidence for mitochondria and chloroplast being from other organisms, right? So we're gonna skip that one. I wanna move on to this one. All right, name one phase of mitosis and explain what happens during that phase. Briefly. You can give one word answers, it's fine. I would prefer that it, obviously this one have to be at least two. Velocity phase and what it does. So yeah, hint, hint, it's gonna be something phase for the name of these. There is, yeah, there's a word limit. Like, we've still got 40 seconds. So you're good.
Bad enough. Miss Dr. Shear's peanut.
Yeah, I like it. The puppy pees on the mat. Pea mat. All right, so we were to name one phase of mitosis and briefly explain what happens during that phase. All right, so we've got anaphases over here, anaphase, metaphase, 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 telophase, telophase, prophase, close, paraphase. It'll be prophase. Could be autocorrect there, right? Autocorrect. It's just the friend of everybody. And then anaphase. All right. So we have all of these. All right, so, which of these groupings do we feel like has the best groups of answers, I guess we could say, but just really quickly vote for them. This part isn't as important. It'll have you like click through those 4 groupings that we just made. I feel like, if I'm being honest here, I feel like prophase got the most underrepresented here. So prophase is our prep phase while you guys are voting. Prophase is our kind of prep phase. Metaphase, lining our chromosomes up in the middle to get ready to separate them. Anaphase, separating the chromosomes. Telophase, separating the cells out into final cells. Pretty good explanation here. Great job, guys. I actually agree. I would have picked metaphase explanations myself if I were choosing voting. I feel like you guys did pretty good with your explanations for that. Some of you guys got some great ones here and the other ones too, though, so. All righty, next one, we're gonna do one more and then we'll just call this one quits. How do the nucleus, endoplasmic reticulum, and Golgi apparatus work together to make cellular products? So this Kahoot is posted online. It's similar, set up similar to this, and I won't be going through and grading those Kahoots, but you are welcome to complete them to test your knowledge, okay? So if you guys want to go online and complete the rest of the questions that we didn't go over for this Kahoot, I have that posted in that discussion reply area, and you guys can go through the rest of the questions that we didn't get to today. That would help you to review that. And then we will have to go up to the lab after this. We got Graham stains today. All righty, we ready? All right, how do the nucleus, endoplasmic reticulum, and Golgi apparatus work together to make cellular products? The nucleus contains the genetic information. It's like the blueprints for everything. All right, then we would make a copy, a working copy, take it to the endoplasmic reticulum. And the endoplasmic reticulum will actually make the protein that that gene is coding for. And then that'll get moved to the Golgi apparatus, where we will package it and transport where it needs to go. We'll also make any modifications that might technically need to be made on it before we go through that shipping process. So here we can see some people, they produce, package, and transport proteins. That's pretty much the steps that we would be following there. But here we have nucleus, DNA replication, and then we will have moving to the outside, the ER ribosome is studied, helps with process along. Golgi apparatus, I don't remember fully. That is fine. It's our transport and packaging thing. We won't read through all of these, but it seems that you guys are getting the gist of this, right? Nucleus, that's our basically codes for the protein. ER, making the protein. Golgi, packaging and transporting the protein. Okay. All right, so we will call that a quits for today. It does have like a leaderboard and stuff like that, but we didn't get through enough questions to bother with it. So we'll just move up to the lab. You can keep those for yourselves or throw them away. I don't care, whatever you want to do with them.